Hi, my name is Ian, and today I'm going to talk about the dendritic cell. Dendritic cells are a type of antigen-presenting cells that play a crucial role in the immune system. They have long tentacle-like arms called dendrites, which allow them to effectively capture and process antigens from pathogens such as bacteria using enzymes. Once they take up these pathogens and break these antigens down, they present them using a special molecule called MHC class I or MHC class II. Dendritic cells will move to the lymph node, where they present the broken down antigens to T cells, activating them and initiating an immune response. Just like the most innate immune cells, the dendritic cell starts from the hematopoietic stem cell, which developed from the bone marrow. Then the hematopoietic stem cell will undergo a process called myelopoiesis, which turns the hematopoietic stem cell into myeloid progenitor cells, which then further differentiates into monocyte and finally turns into cells like the macrophage and the dendritic cell. Because the dendritic cell is an antigen-presenting cell, it has a variety of receptors on the cell surface. Some of the most important receptors on the cell surface include the mannose receptor. This interesting receptor can recognize a variety of carbohydrates on other cells, but it mostly helps with endocytosis, which behaves similarly to macrophages. The second receptor is the DC sign receptor. This receptor highly recognizes high mannose type N glycan, which are commonly found in bacteria. The third major receptor that I'm going to talk about on the dendritic cell is the scavenger receptor. This receptor helps to locate and binds to multiple different molecules that are found in bacteria. Lastly, there's the toll-like receptors. These receptors act very similarly to pattern recognition receptors, but instead it recognizes structurally conserved molecules derived from microbes and helps create an immune response. These variety of receptors allow the dendritic cell to become more diverse and adaptable to many different solutions. When the dendritic cell enters the battlefield, the first thing it will do is to start rapidly uptaking nearby bacteria, using endocytosis and then breaking them down using special enzymes to turn them into antigen fragments. After the dendritic cell consumes enough bacteria, it will quickly move to the lymph nodes, where it will then present the antigens on special molecules called MHC class I or MHC class II, and then show the antigen fragments to T cells. This entire process takes around a week or less to activate and stimulate the T cells. Then activated, T cells would rapidly duplicate and go back to the battlefield, where it would start to rapidly kill the recognized pathogens. The dendritic cell would then return back to the battlefield to capture more bacteria, and this process repeats. The dendritic cell processes antigens in 4 main steps. First, the antigen will be grabbed and then taken up using endocytosis and then put inside the dendritic cell. Second, the bacteria, after being taken up, special enzymes will break down the bacteria and turn it into antigen fragments. Step 3. The dendritic cell will use special molecules called MHC class I or MHC class II to present the broken down antigens. Finally, step 4. The dendritic cell will present the MHC class I or II on its dendrites to activate specific T cells. The last thing I'm going to talk about is the MHC class I and MHC class II. The 4 main differences between these two molecules include number 1, the shape. As you can see, the MHC class I has 1 long segment, 1 medium-sized segment, and 1 short segment, but the MHC class II has 2 long segments. The second main difference between the two molecules is that MHC class I can be found on most nuclei cells, but the MHC class II can only be found in specific antigen-presenting cells, like the dendritic cell and macrophage. The third main difference is that the MHC class II is able to present longer antigens fragments due to its structure. And finally, the fourth main difference is that the MHC class I activates CD8 T cells, while the MHC class II activates CD4 T cells. All the components I just listed makes the dendritic cell one of the most vitally important cells in the immune system.